I would like to introduce the food metabolomics research conducted in the Fukusaki laboratory. Food metabolomics is a field of study that applies the metabolomics method, which analyzes components to the food sector. From raw materials to the food we eat, food goes through many processing steps. These steps, such as storage and cooking processes, can affect food components, ultimately reflecting differences in sensory attributes and perceived food quality for consumers. Foods contain various components, which are intricately related to qualities such as taste and aroma. To clarify the relationship between food quality and its components, we conduct research using metabolomics techniques to analyze a vast array of components. Here, I would like to introduce one of the frameworks used in food metabolomics. For food samples, both sensory evaluation by humans and component analysis using instruments are performed. In sensory evaluation, qualities such as taste and aroma are assessed. In instrumental analysis, components such as sugars, amino acids, and alcohols contained in the food are analyzed. The data obtained through these methods are then analyzed using statistical approaches such as multivariate analysis to explore the relationship between food quality and its components. Discriminative analysis in metabolomics is used to investigate differences between sample populations without obtaining detailed information about the samples. On the other hand, informative analysis in metabolomics can characterize and identify target compounds to search for novel bioactive compounds, biomarkers, and to create a metabolite database for specific food samples. Metabolomics is applied to a wide range of foods, including agricultural products and fermented foods. In the Fukusaki laboratory, we conduct research on various foods, including fruits, coffee, chocolate, and tempeh, a fermented food from Indonesia. Through component analysis, we aim to manage and improve the quality of food and identify its origin. Now, I would like to introduce some of the actual research being conducted. The first topic is the study on predicting the ripening stages of fruits. Bananas are known for ripening after harvest, during which their taste and color change. The color change in the fruit is used as an indicator of its ripeness. In this study, the relationship between the color of the fruit and its components was examined. First, the components and color of fruits at various stages of ripeness were analyzed. By clarifying the relationship between the components and color, it became possible to predict the color based on the components. The results of this study are expected to contribute to improving the ripening process and post-harvest preservation technique. Not only fruit ripening, but studies of different cultivars of varieties of fruits can also be performed. For example, in this lab, we also conducted component profiling of mangoes from different cultivars. In collaboration with an Indonesian research agency, we applied a metabolomics approach and sensory evaluation to identify the characteristics of these mangoes. The data obtained can be used for discriminative analysis to identify unique attributes. The next study is on metabolomics for tempeh. Tempeh is an Indonesian fermented food made by inoculating fungi into cooked soybeans. It is similar to natto but has a solid texture. Tempeh is rich in protein and is an important food that can serve as meat alternatives. It is gaining attention worldwide, particularly from vegans and vegetarians. Tempeh is made by peeling, soaking, and boiling soybeans, followed by fermentation using fungi. However, the methods of tempeh production vary by country and region, with some places using fully automated systems, others using semi-automated systems, and some still relying on traditional methods. Due to differences in production processes and variations in the raw materials, such as soybeans and water, the compositions of the resulted tempeh are diverse. Metabolomics is used to investigate the differences in tempeh production methods and regions. For a total of 17 samples made using different production methods in different regions, component analysis was conducted using a gas chromatography mass spectrometer. First, principal component analysis revealed that the samples clustered based on the differences in production methods. Additionally, by using a color measurement device to analyze the surface color of the tempeh, it was found that tempeh produced using traditional methods had a darker color compared to those produced using semi-modern methods. From the component and color data, we were able to identify components that are likely involved in the color intensity. One of the most interesting studies is coffee, because many people love coffee. To determine coffee quality, there are standard procedures called cupping tests that conducted by trained and certified panelists. If the coffee is scored more than 80 out of 100, this coffee being categorized as specialty coffee. And to know what are the key compounds that correlate with coffee quality, metabolomics is a powerful tool to answer these problems. Coffee is analyzed using a gas chromatography mass spectrometer to obtain component information. At the same time, a cupping test is conducted by experts to obtain sensory evaluation scores. By combining these results, potential compounds important for quality can be identified. These potential important compounds were then added to coffee to investigate whether they would improve their sensory evaluation scores. Finally, the study focuses on exploring compounds that are correlated with the taste and characteristics of coconuts. Coconuts are a typical tropical fruit widely used in Southeast Asia, with various parts utilized for different purposes. Currently, using metabolomics techniques, research is being conducted on the preservation of coconut water quality, the identification of the special cultivar copior, and the impact of origin and cultivar. By investigating the correlation between metabolite profiles and physicochemicals and also sensory profiles, analyses are being performed to understand the effects and identify the characteristics. These are the examples of publications by the food metabolomics team of Fukusaki Laboratory in 2024. Many papers have also been published, so if you are interested, please feel free to contact us. That's all. If you are interested or have some questions, you can come to Fukusaki Lab directly. Thank you for your kind attention.